If you're looking for a solid overview of the process of transcription, stick around. We'll get you understanding what's going on. Hi biology friends, it's Sarah, your other biology professor, and let's talk about transcription. In another video, we look at some differences between how bacteria and eukaryotes transcribe. But here, we're going back to basics with what everybody has in common. In transcription, we're using DNA as a template to build RNA. It's a critical step in gene expression, the process of going from DNA to a functional product. It's how we get from a genotype, what the DNA says, to a phenotype, a physical or physiological characteristic that we can observe. So DNA as a template. Which DNA are we using as a template? Genes. Genes are regions of DNA that are transcribed. Genes have distinct structures. The exact details will vary by the gene and the organism, but a gene will have a promoter region for setting up transcription, a transcribed region that will be read, used as a template to make RNA, including some signals for termination that determine where to stop. What kinds of RNAs are we building in transcription? Any RNAs. We often focus on transcription of messenger RNAs, mRNAs, as part of protein synthesis, but other types like tRNA and rRNA are also made by transcription. We're just making a single-stranded RNA polymer here, regardless of the type. RNA is similar to DNA in many ways, but there are key differences in structure, like RNA nucleotides use a different sugar, and the base uracil instead of thymine. When you're building important molecules in molecular biology through complex processes like DNA replication, transcription, and translation, you'll often see those processes broken into stages: initiation, elongation, termination. Let's see what happens in each stage. Initiation is our setup phase. We're going to gather the materials, the proteins that we need for transcription, and get them set up in the correct location. Okay, so what's the correct location? Well, that's where the promoter comes in. It's our staging area. The promoter is a non-coding region of DNA. That means its sequence does not encode protein. The promoter is located upstream or before the transcribed region, and it has specific sequences for protein binding. These sequences vary based on the organism and the gene, so eukaryotic promoters look very different from bacterial promoters. These promoter sequences determine where transcription begins, called the transcription start site. All right, so what proteins are going to bind the promoter? Well, the enzyme that joins RNA nucleotides to produce RNA strands is RNA polymerase. Bacteria have one type of RNA polymerase. Eukaryotes use at least 3 for different types of RNAs. But RNA polymerase can't find the promoter sequences on its own. It needs some extra proteins for help. Proteins called transcription factors recognize promoter sequences and bind first, then allow RNA polymerase and sometimes other proteins to bind. We can't initiate transcription without these guys. Other DNA regions outside of the promoter might serve as regulatory sequences. More transcription factors will bind here to influence how much transcription is happening, either increasing or decreasing gene expression. Once it's on the promoter, RNA polymerase, or one of its associated proteins, acts as a helicase to open up the DNA to form a transcription bubble. Now the DNA can be read, but only one DNA strand will act as the template strand for transcription. Which one? The promoter tells us that too. The sequences that proteins bind are on the non-template strand. RNA polymerase will use the other strand as the template, adding the first RNA nucleotide complementary to the DNA base in the transcription start site. C's and G's pair, T in DNA pairs with A in RNA, and A in DNA pairs with U in RNA. In elongation, we're building our RNA polymer. growing the nucleotide chain through complementary base pairing. The most recently added RNA nucleotides will be inside the transcription bubble as part of a DNA-RNA hybrid created by base pairing. What types of bonds are formed here? Phosphodiester bonds connect a new RNA nucleotide to the growing RNA strand, and hydrogen bonds temporarily form between the DNA template and the complementary RNA nucleotides in the hybrid. Like with our other molecule building processes, directionality is important in transcription. RNA polymerase works in one direction. It can only add new nucleotides to the 3' end of the growing RNA strand. That means that RNA is produced 5' to 3', we add to the 3' end. So to do that, RNA polymerase reads the DNA template strand in the 3' to 5' direction. Notice that the RNA sequence is complementary and antiparallel, opposite 5' and 3', compared to the DNA template. But the RNA sequence is the same as the sequence on the other DNA strand, the non-template strand, except for T's and U's. And the directionality is the same. And so the non-template strand is also called the coding strand because it shares the same code as the RNA. You'll also see it's called the sense strand. So RNA polymerase keeps reading the template strand and adding RNA nucleotides to the 3' end of our RNA strand that are complementary to the DNA base it reads, and then opening up more transcription bubble as it moves until the end. But we're not just transcribing all the way until the end of the chromosome. Instead, signals will determine where to stop. A termination signal marks the end of a gene and triggers termination of transcription. This process may require extra proteins to get involved and interfere with RNA polymerase. It depends on the gene. In termination, RNA polymerase is going to be dislodged from the DNA template. The DNA-RNA hybrid breaks apart, and the RNA molecule, or transcript, is released. Now, depending on the organism and the type of RNA that we've just made, that transcript may need some modifications before it can be used. In bacteria, transcription and translation happen in the same space, the cytoplasm. So ribosomes often hop on an mRNA in progress and start making protein while that mRNA is still being transcribed. In eukaryotes, the processes are separated. Transcription happens in the nucleus, where the DNA is, and mRNAs have to be completed, modified, and exported to the cytoplasm for translation. All right, let it all soak in. And if you're ready and willing for even more transcription, I've got a few videos to recommend. Meanwhile, give me a thumbs up if transcription makes a little more sense now. Subscribe for more biology content from me. Check out my website for tutoring resources, and I will see you next time. Bye.